Good morning, my dear students. In the previous class, I was discussing something about the importance and the need for the food production. India is a very thickly populated country and it is also supposed to be a poor country. We have to feed the mouth of all the people with the existing small amount of food that we are able to produce. Now, when the population is increasing day by day, the scientists in India are trying all their level best to increase the production of the food. So, food production has become as a very important topic, or it has become one of the important problems of the nation. How the food production could be improved? Using only the limited amount of the land that we are having is the primary concern of the people who are in India today. How this is taking place, a part of that has been completed in my previous class, and today it's going to be just a continuation of what I was discussing in my last class. I told you that the food production could be increased by many methods. There are so many methods for that. One very important method is selection. What is this selection? You select only the good crops. See? Even Darwin has told survival of the fittest. So that is a basic concept. That which is fitting in this society will be able to survive better. So, you select only the fitting elements. Then, with that elements, you will be able to improve the quality of the crop. So, this is the basic concept about that. Okay. So, you select the better crops, the best crops. And then with the help of those crops, you try to produce the progeny. Then, the food production could be improved by this method. And this is once again divided into three categories, namely mass selection, pure line selection, sorry for the interruption. So, this selection is of three types, namely mass selection, pure line selection and pedigree selection. So, by these three methods, we can improve the quality of the crops. Then, I told you that plants could be, or the food production could be improved by means of what is called as introduction. Now, what is called as an introduction? The different food crops are introduced from different parts of the world, and then the food wealth of our country could be enriched by that. The Britishers, when they came to India, they brought a number of crops with them to lead their type of life in our country. Similarly, when the Mughals came from their country to rule us, they brought their own food with them. Portuguese came. Like that, when different people came to India, they brought their own crops from the different parts of the world. And in fact, from botanical point of view, it was good for us because we were able to enrich our food content of our country. Then, hybridization is another method by which you can improve the crops. Now, these are the methods that we have analyzed so far, and we are going to proceed for the other methods. So, it is coming under the plant breeding part 2, by which we can improve the crops. So, hybridization is a method in plant breeding to improve the native crops by obtaining diverse genotypes that can be used as a source material for collection of crop with desirable characters and genes obtained from many parts of the world. So we get different crops from the different parts of the world, and then we do the hybridization. When you cross two plants, the characters derived from the two plants could be pooled or could be put in one plant, and that plant will be a successful survivor, and you can derive all the benefit, good qualities from that particular plant. So hybridization is one of the methods by which you can improve the quality of the crop. Then this hybridization is done by different methods, that you take two plants and then you cross. But when it is not, when there is no compatibility, when you are not able to cross the two plants, then you go for what is known as a protoplasmic fusion. So a hybrid produced from the fusion of a protoplasm of two different species is called somatic hybridization. So it is a branch of hybridization, different method of hybridization. When you take two plants and then make a cross, it is called simply a hybridization. When there is no compatibility between two plants, particularly for example, when you take a plant A, plant B, and if they belong to two different species, for example, one is a potato, then another one is a tomato. Potato is called as Solanum tuberosum, technically binomial. Tomato is called as Lycopersicon versicolor. Now they belong to two different genera. For example, potato is belonging to the Solanum genus, and this is belonging to the Lycopersicum genus. The number of chromosomes may be the same, but still the two chromosomes, they belong to the two different groups. So there will be less compatibility or no compatibility between these two plants. Scientists wanted to bring both the characters in one plant. A plant which could have potato at the base and a tomato at the top. So when you are trying for a hybridization between the two, it may not be possible because of incompatibility. But here, protoplasmic fusion they are trying. You take one cell from this potato plant and another cell from the tomato plant, and then you try to cross these two cells instead of two plants. What they do, they follow a different method. The cell is taken and then it is burst by a suitable technique, and here also it is done. Then the nucleus of this one is made to fuse with the nucleus of the other one. So artificially you are compelling, in fact it is a matter of compelling. You are compelling the two nuclei to fuse together to produce or to become a different type of nucleus. In fact, it is in that way how to tell. So this is what is called as a protoplasmic fusion. You fuse both the protoplasm. A hybrid produced from fusion of a protoplasm of two different species is called somatic hybridization. It could be two different species or it could be two different genera. Fusion of a protoplasm from two different varieties can be enhanced by treatment with the chemical called polyethylene glycol. Polyethylene glycol, simply called as a PEG, PEG, in the presence of a high voltage electric current on a suitable medium. Now, by this method, somatic hybrid plants with desirable changes can be obtained. This method in plant breeding is called as a protoplasmic fusion. So when natural hybridization is not possible, they follow this protoplasmic hybridization, or in other words it is called as a protoplasmic fusion. Now there is another method of a crop improvement or a food production, better food production, is tissue culture. Many important food plants like tomato, banana, apple, etc. have been produced on commercial scale using this method. So you take a single cell and then by suitable culture, by culturing this single cell, it could be made as a callus, and from that an organ could be produced, and from this organ a young plantlet could be produced, and that plantlet, when it is taken to the garden or ground natural situation, you harden the plant, and then from that one you make a new plant to come out. So by the tissue culture techniques you can produce new plants in this world. So this is used as one of the important methods for crop improvement. Now what is heterosis? The superiority of the F1 hybrid in performance over its parent is called heterosis or hybrid vigor. Vigor refers to increase in growth, yield, resistance to diseases, pest and drought. So the idea of or the heterosis is what is heterosis? It is the superiority of the F1 generation over its parent. So there is a plant A and it has got certain qualities. You cross it with B with certain qualities. Now when you cross these two plants, you get a plant C. This plant C is superior over A and superior over B. Now this term superior is very vague in science, and when you say a plant is a superior with reference to the parent, in what way it is a superior? So this superiority could be in any form. For example, yield will be more. The size of the fruit or the seed, size of the fruit or the seed or even the tuber, that could be better. Then there won't be any difference between A and C, but C will be a disease resistant plant or disease resistant variety, what we call as a DRV. Sometimes you could be able to resist the drought. So drought resistant variety, it could be a drought resistant variety also. It could be a saline resistant variety. So the superiority can be in any method or by any way. Okay. So it need not be only with respect to the yield. It could be in with reference to some other good character also. So the superiority of the F1 hybrid in performance over its parents is called heterosis or hybrid vigor. Vigor refers to increase in growth, yield, resistance to diseases, pest and drought. F1 hybrids of maize show 21% increase in yield when compared to their own parent crops. Vegetative propagation is the best suited measure for maintaining the hybrid vigor. So once a new crop is produced, so as I was telling you, when A is crossed with B, then by the hybridization you get a crop C, and you find that this crop C is definitely superior over A or B, then this can be further taken to the subsequent generations by vegetative propagation. Because if you are going to cross this plant with some other plant D or E or any other plant for that matter, then the chromosome contamination will take place and this superiority will be lost. So once you think that you have produced a superior crop over the parental plant, after that it could be taken to the subsequent generations only by the vegetative propagation, because when you do a vegetative propagation there will be no contamination of the chromosomes. So F1 hybrids of maize show 25% increase in yield when compared to its own parents. The vegetative propagation is the best suited measure for maintaining the hybrid vigor, because once a hybrid is produced and if you are going to cross it with some other plant, this hybrid vigor will be lost. Heterosis or hybrid vigor. When two species are crossed, F1 progeny is superior to parents. Vigor refers to increase in growth, yield, resistance to diseases, pest and drought. Example, F1 hybrids of maize show 21% increase in the yield. Vegetative propagation best suited for maintaining the hybrid vigor since the desired traits can persist over a long period. Here you see, this is an inbreeding. In a particular plant you have got both the you have got flowers and each flower is bisexual and you do inbreeding. But different flowers of the same plant, when you cross the hybridization or when you cross it with other plants, it becomes hybridization. Now these are the two parental, this is the corn of the parental varieties. And you see the corn producer, when you make a cross between these two varieties, you see how it has. Definitely this seems to be a better crop with respect to the yield. Now you just see this plant and this parent. When you cross these two plants, you get a plant which is of superior nature, lengthwise, then production wise, and in most of the characters, most of the qualities, this crop will be better over the parental varieties. Now some more examples for the better plant or a better variety produced from the parental variety is a triple gene dwarf wheat. This is a variety, triple gene dwarf wheat. It has got increased height and yield. Then second example is Atomita 2 variety. It is a rice variety with saline tolerance and pest resistant. So it has got two good characters. One is a saline resistant character and other one is a pest resistance or pest tolerance character. So two good qualities or two hybrid qualities it is having over the parental varieties. Then example 3 is groundnuts with a thick shell. So that's very important. It has got a thick shell over it, so it won't get easily contaminated or easily broken. So these are the three examples where the F1 hybrid is better over their parental varieties. Very good examples for the heterosis. Genetically engineered hybrid plants you are having. You have probably eaten many genetically engineered plants without realizing that it is a genetically engineered variety. Without our knowledge we are doing that. And the examples include seedless grapes, best example for a genetically engineered crop. Then watermelons, which is also seedless. Bananas, which commonly have polyploidy, that is extra set of chromosomes which is having the stronger and bigger. Then corns, other very good examples. Modern corn was selectively bred from Josephine at least 6,000 years ago, they are telling. Okay. So these crops are in the usage of man for a quite long time, and consistently man is improving the crop by the different hybridization techniques, and these are all some of the plants that we are regularly using by genetically engineered techniques: seedless grapes, watermelons, bananas, and then of course the corn. So these are some of the crops which are genetically engineered to give better result for our food production. Now you see some more crops which are genetically engineered and then modified, which we are using in our day-to-day life. One is a soya bean, and then corns, then rice. This is also genetically engineered or genetically modified, different varieties of rice we are having. Canola oil that we are using in our day-to-day life in our kitchen. Tomatoes, potatoes, cotton seed, then papaya, then peas, and then dairy products that we are using today. Of course, these dairy products are, it's an animal example, but all others are the plant examples. So these are all some of the engineered, genetically modified plants, genetically with better vigor, and then the production will be better and yield will be more. Commercial, they have been commercialized totally, and then because of this commercialization, people are able to get more benefit, and as a user, we are also benefited because the yield will be better, it will be cheaper. When it is produced in a huge quantity, they are able to reduce the price also. So it is beneficial on both the sides. So these are all some of the examples for genetically modified crops. Then polyploidy in plant breeding. Now we will just tell you what is a polyploidy, and then we will go to the other areas. Then it will be easy for you to understand. Now when you get two pairs of, two sets of chromosomes, it is called as a diploidy. Now I have taken a plant with just 4 homologs. So 2n number is equal to 8 here. Now if you add one more set of chromosome, then now it becomes a triploid crop. If you add one more set, then it becomes a tetraploid crop. So more than 2, it is a polyploid. When you get only 2, it is a diploid, 3 triploid, then tetraploid, pentaploid, hexaploid, heptaploid, octaploid, nonaploid, decaploid, dodecaploid, like that it goes. And you have got chromosomes up to, even in some plants you have got 12n chromosome number also we are getting. 4n number tetraploid is very common in the plants. 6n hexaploid is also very common in the plants. Polyploidy is not common, or it is not possible to that extent in the animals. Only few animals are there with the polyploid nature. But in plants, it will be very surprising to know that most of the food crops that we are using today, they are only polyploids. Okay. Now what happens when a plant is with only 2 sets of chromosome like this? It is having some effect. It is producing a crop with a particular size, and its tuber content is with a particular size. Or if it is an oil yielding crop, the oil output will be in a particular level like that. But when you are increasing the number of the chromosomes to triploid or a tetraploid, etc., now what happens? When more number of chromosomes are there, the output will also be more. So naturally a polyploid crop will be able to give a better yield. So by increasing the chromosome number, by making a plant to become a polyploid, the quality or the yield of the crop could be maximized, or it could be made better. So when you increase the diploid number to a tetraploid or hexaploid, then the yield will also be better. So there is a separate topic called breeding the polyploids for the crop improvement. Okay. Now, what happens? You take two plants which are diploid in nature. Let us imagine this is a plant A and this is a plant B. Now the chromosome will be different because this is a plant A, this is genus A and this is a genus B. Now let me draw this diagram in this way. This is a diploid plant and this is a diploid plant. Now what is happening when this is undergoing the pollen genesis? This will be the type of the chromosomes derived from this because it will undergo reduction division. This will be producing the genes in this way. Now what will happen? This will not be able to, this is a diploid plant having the chromosomes from both the plants, but it cannot undergo meiosis in the subsequent generation because it will not be having the compatibility. This chromosome will not be able to pair with this chromosome. So meiosis cannot take place in this chromosome. So this diploid can be propagated only by the vegetative mechanism, even though it is having the good characters between the two. So what they do at this stage, they induce a polyploidy, or they induce what is called as a colchicine. When you add a colchicine, this becomes a tetraploid. That means a plant of this nature will be produced. Now what will happen? See, the chromosomes will arrange themselves in the equatorial plane like this at the time of a meiosis, like this. So they will try to pair among themselves and they will try to pair among themselves. So the pairing will be taking place in this way. This will go to one pole and this will go to another pole, and so the perfect meiosis will take place. Absolutely there won't be any problem. So when you take two unrelated plants called A and B, and when there is no compatibility, when you compel the plants to undergo fusion, and even when it is a producer, an F1 plant is a producer like this F1 plant will be having the benefit only on that generation. It will be a sterile plant. It can't produce any flowers. Why it is not producing any flowers? Because meiosis cannot take place in that. When meiosis is not taking place, a pollen grain germination formation is not possible, ovary formation is not possible. So the F1 will be having all the good qualities, but still it is a F1 plant will be a sterile plant. So it is an F1 sterile variety. Now when this F1 sterile variety, if you add colchicine, what will happen? It will become a tetraploid, but this tetraploid will be a fertile variety. So polyploid is used for for even introducing the what is called as a fertility in the plants. So polyploid breeding, how it is done? In nature, heritable and desirable variations occur by polyploidy mutation, recombination and chromosomal aberrations. Any organism with a 2n number of chromosome is doubled and it is called as a polyploid. Polyploidy can be induced by use of a colchicine, which is a plant product. It is taken from a plant called colchicum autumnale. It is extracted from a plant called colchicum autumnale belonging to the family Liliaceae. Okay. So this colchicine is taken and it is induced. Now, the induction of a colchicine is a separate topic. It can be inducted or introduced in the bud, or it could be induced in the seed. There are different methods of inducing polyploidy by using the colchicine. That's a separate topic. Those who are specializing on polyploidy breeding and those who are really involved in the plant breeding techniques, they will know the different methods of inducing polyploidy by using colchicine, and it is not required at your level. How the colchicine is induced into the plant, or it is administered into the plant to have a polyploid plant. So these are all some of the main crops which are of a polyploid nature, as you see, as I was telling you. A hexaploid plant, a tetraploid plant, this is also a tetraploid, a triploid plant, a heptaploid plant, and then octaploid plant. So these are all what is called as a polyploidy is a very common in the plants, which is of a very rare nature in the animals. So a common wheat is having 6n number. Tobacco, which we are using today, is a tetraploid. The potato is also a tetraploid plant. Banana is a triploid plant. And then basinberry is a heptaploid plant. Strawberry and octaploid plant. So polyploidy is a very common feature, and when you are increasing the number of the chromosomes, it will be giving a better result. But unfortunately, this you don't have a, fortunately or unfortunately, you don't have a polyploidy in a man, human being or animals. Even if one chromosome is increasing or decreasing in human being, what effect it is having? You know that you have studied in zoology. If it is a 21st pair of chromosome and one chromosome is just increasing, it is called a 21st trisomy, and it is a Down's syndrome. It is giving rise to so many problems. The chromosome in the sex chromosome, when there is an increase polyploidy, the normal chromosome, if one X chromosome is more, it is called as a Klinefelter syndrome. And if one chromosome is lesser, you got only no Y chromosome, that Y is lost, only one X chromosome is there, it is a Turner syndrome. So different type of syndromes are produced even when there is one chromosome extra or one chromosome lesser. When that is the case with the animals and human being, so that's not a problem for the plants. When there is an increase and decrease in the number of the chromosomes is not much a problem for the plants. In fact, increasing the number as a set, as a triploidy, tetraploidy, hexaploidy, etc., they always give a better yield, better result. But only thing is, as I was telling you, sometimes the F1 hybrid is becoming sterile. For example, this 3N banana will be a sterile F1 plant, because when you are having 3 sets of chromosomes like this, a triploid plant, meiosis cannot take place. Meiosis cannot take place. When it is a diploid plant, one set will go to one pole, another set will go to another pole. So a triploid plant can never be a fertile plant. It could be only a sterile plant. So if it is made what is called as a hexaploid, now it is possible, because these newly formed cells will come and arrange in a different plane, and it will be pairing in this way. So 6, 24 I have drawn. These 24 will be arranging 4, 5, 6, 7, 8, 9, 10, 11, 12. Now these 24 chromosomes will come and arrange like this in the equatorial plane, and then it can be meiosis could be formed. So a triploid plant, when it is made as a hexaploid, then fertility could be introduced into that. So polyploidy, that it is helping in overcoming the sterility of the plants. அது அட்டெண்ட் பண்ணுங்களா. Now Mutation in plant breeding. The variant plant which suddenly produced large and bright colored seeds due to mutation was selected and subjected to inter or intraspecific crosses with our native crop. Thus, new varieties are produced and released. For example, Mungo 1 variety is cultivated in Punjab. Okay. So, next area is what is called as mutation breeding. What is a mutation? When you get a pair of chromosome and when you have got the genes inside, you know the genes are expressing in the form of protein production, oil production or something like that. Now, when you are able to change the nature of that gene, you try to locate which gene is having what effect. What are the genes which are responsible for the oil production? Which genes are responsible for the protein production? So, you try to locate that gene, and then when you are manipulating that gene, it will be able to give you a better result. So, this is what is called as a mutation breeding. When you take now, how the mutation breeding is done, you take a field and in this field thousands of crops are here, okay, and this field is subjected for mutation. So, what they will do, they will erect a pole in the center of the field, they will erect a pole in the center of this field. So, and then from the top of that pole, X-ray emitting unit will be there, or there are different methods of inducing the mutation. The whole thing will be completely fenced to prevent the radiation to the common public. So, it's all done very carefully, extremely carefully. So, they will induce the mutation and the seeds will be collected from this and it will be called as a M1 generation. Just like a back cross a B1, B2, B3, you have got a M1 generation. This M1 generation mutation for first crop. So, out of these 1000 plants, some plants will be able to give a better result. Definitely, you have to look for it. You have to collect the seeds and then you have to sow the seeds, and which is showing the better quality, a better variety, you have to select. Then the better plants that you have selected from the mutation 1 could be made to taken to subsequent generations, namely M2 generation, M3 generations, etc., etc. So, when you expose the plant for radiation, see, it is you can't say that a mutation is always dangerous. A mutation will give a better result also sometimes. So, you have to look for it. Then it becomes a beneficial one. Even the mutation becomes a beneficial. So, mutation is the process by which genetic variations are created through changes in the base sequence within genes, resulting in the creation of a new character or trait not found in the parental type. It is possible to induce mutations artificially through the use of chemicals or radiations like gamma radiations, and selecting and using the plants that have the desirable character as a source in breeding. This process is called as a mutation breeding. Example, mung bean we have produced as a result of the mutation breeding. Now, so these are all some of the examples, beautiful examples of the plants which have been produced by what is called as a mutation breeding. The fertile twin grain mutant, it has 2 sets of pistils suggesting better external pollen reception. This recessive mutation will not be expressed in the F1 generation. Then, flowers of a twin grain mutant has 2 pistils, both are fertile if a condition allows. These characters expressed in the maternal tissues may be screened in the M2 generation field. M2 generation field. That is, in case of a twin grains, flowers in mutation 2, second generation plants, and seeds shown above are in the third mutation generation. In rice, the flowering time of the day is not long, a few hours in the morning. This open hull mutation keeps open its flowers or cannot dose. However, this mutant has a good seed fertility as shown in the picture. So these three mutant crops have got a better result over their parental varieties. Now we have got another very beautiful interesting topic. Sometimes you breed the crops for the disease resistant. Now we find that in what is called as tribal varieties, in country varieties, in the traditional varieties, the plants are there, and these plants, let's say these are all the traditional varieties, the yield will be very less. Normally we find that the yield will be less, but it will be having the disease resistant genes will be there in these varieties. See, this common feature you could have seen in the human being also. When you see some of the tribal people, they won't be much caring for their health. They will be doing what all they want, but still their disease resistant character will be there for them. So they have got a better disease resistant characters. So these disease resistant characters will be there in the tribals. So like that in the tribal varieties, you will be able to have the disease resistant genes will be there. What you are doing, you take those tribal varieties, and from that you separate the disease resistant character, or by crossing techniques you can bring the disease resistant genes into the modern crops. So the modern crops will also be having the disease resistant character. A wide range of fungal, bacterial and viral pathogens affect the yield of the cultivated crop species, especially in tropical climates. Crop losses can often be significant, up to 20 to 30%, or sometimes even total. In this situation, breeding and development of cultivars resistant to diseases enhances food production. This also helps to reduce the dependence on use of fungicides or bactericides. So if a plant is attacked by a disease, then you will be having, you have to apply fungicides or you have to apply the bactericides. But if you are having a what is known as a disease resistant variety, disease resistant for fungus and disease resistant for bacteria, then you need not use. So the cost wise it will be very beneficial for the farmers. These disease resistant, that is a fungal resistance, and a bacterial resistant varieties will be of much useful to the farmers, because they need not spend any or much money for these diseases. The various sequential steps are screening germplasm for resistance of the sources, hybridization of selected parents, selection and evaluation of the hybrids, then testing and release of the new varieties. So these are the different steps involved for the disease resistance. See, when a crop is, see how a disease has attacked a tomato, and then see the condition of this tomato plant, how much damaged it is. Plant breeding for developing resistance to the insect pest. Breeding methods for insect pest resistance involve the same steps as that for the disease resistance, and any other agronomic trait such as yield or quality and are discussed above. Sources of resistance genes may be in cultivated varieties, germplasm collections of the crop, or wild relatives. Now, what is biofortification? Breeding the crops with higher levels of vitamins and minerals, or higher protein and healthier fat, is the most practical means to improve the public health. So the crops produced by some mechanism will be having more vitamins, and then healthier fat will be there. So if you are able to produce a crop with these qualities, then definitely it will be able to improve the public health also. So this is what is called as biofortification. Breeding for improved nutritional quality is undertaken with the objective of improving the protein content, then oil content, then vitamin content, and then the micronutrient and mineral content. So if you are able to improve the content of these things in a plant, then that is what is called, that is going to help in the improvement of the public health, which is called as a biofortification. Then what is called as a SCP, single cell protein. There are certain algae which are able to give a better result, and so these are highly proteinaceous in nature, and that goes by a separate topic called the SCP or a single cell protein. Microbes are being grown on an industrial scale as a source of a good protein. Microbes like spirulina can be grown easily on materials like wastewater from potato processing plants containing the starch, straw, molasses, animal manure, and even sewage to produce large quantities, and can serve as a food rich in protein, minerals, fat, carbohydrate, and vitamins. Incidentally, such utilization also reduces environmental pollution. So don't think that only the higher plants, angiospermic plants, will be able to give us a food in the form of a paddy or maize or wheat or something like that. Even lower organisms, even algae can become a very good nutritional food, because they are also having very good protein, minerals, vitamins, everything they are having. So these lower forms of plants, they are also very good. They are very good for our health. Now we are going to another very important step called green manuring. When you add a chemical manure in the form of NPK, nitrogen, phosphorus, and potassium, the famous NPK formula, which is the basic ingredient in any of the chemical manure, in a long run this is going to spoil the soil, and the soil becomes an imbalance. Say after 30 or 40 years, the soil will become totally unsuitable for the cultivation, or you have to forget the agriculture in those fields or in the land where you are constantly and continuously using the chemical manure. So to avoid that, nowadays the concept is we have to use the chemical green manure. Crotalaria gentiana, Cassia minisoides, Glycine max, Indigofera linifolia, Thesbania rostrata, Acacia nilotica, Luzina, Athyrus, and Micuna. So these are all some of the plants which are used for green manuring. Most of them are very big trees. Normally branches are cut, and then these cut branches are buried under the soil. This once again is a different topic. How the green manure is formed. You have to form the pits. You have to put the things in the pit. You have to add microbes to that. You have to add salt. Then different water has to be added. Then it has to be kept in that stage for a longer duration. Then it becomes automatically, all the leaves will become the manure. Then you have to dig the manure, and then you have to add it to the plants. So that's another topic of how a manure could be formed from the green plants. So green manuring, they accumulate more than 80 kg of nitrogen per hectare in the soil when grown as a green manures. Azolla is an aquatic fern which contains an endophytic cyanobacterium Anabina azolea in their leaves. So cyanobacterium Anabina azola is there in the leaves, and then that is able to produce or increase the nitrogen content. See, mycorrhiza. Well, in a root system you will be having this, when fungus are growing, it is called as a mycorrhizal association. Myco means a fungus, a rhizome means a root. When you get the fungus growing in the roots of the plant, then it is able to increase the nitrogen content. So mycorrhizal association is another very big topic. There is an ectomycorrhizal association, endomycorrhizal association, and there are certain plants which will never grow without the fungal association, mycorrhizal association. A pinus plant must find a fungus, a mycorrhiza, then only further it will grow. Otherwise, even when it is a seedling, it will die. So the mycorrhizal association is a very important association for the life of the plant. The role of a biofertilizer. Extensive use of fertilizers and chemical pesticides had resulted in the soil and water pollution. Fossil fuels such as petrol and coal are used in the manufacture of fertilizers and pesticides. To reduce the pollution and overusage of our non-renewable resources like coal, petroleum, etc., they are called non-renewable because they are used. Once it is used, means used. You cannot have recycle it. It's a coal. It's used means used. Petroleum means used means it cannot be brought back. It's called a non-renewable. These are non-renewable sources. An alternate method has been successfully developed to safeguard these natural resources. Also, as I was telling you, Azolla is used as a biofertilizer. Azolla is a tiny freshwater fern common in plants, ditches and rice fields. It has been used as a biofertilizer for rice in all major rice growing countries including India, Thailand, Korea, Philippines, Brazil and West Africa. The nitrogen fixing work is accomplished by the synthetic relationship between the fern and the BGA, that is biological gradient of A, Anabina azole. In addition to nitrogen, the decomposed azolla also provides potassium, phosphorus, zinc and iron to the crop. This is a biofertilizer. This is azolla growing in a pond. Benefits from the biofertilizers. Biofertilizers are easy to produce in abundance and are available at low cost to the marginal farmers. It increases soil fertility without causing any damage to the soil. Application of biofertilizers increases yield up to 45%. And leftover biofertilizers in the soil increases yield as long as the biofertilizer remains in the soil up to 3 to 4 years. See, once a biofertilizer is used, it will remain in the soil for a longer duration and it will be helping the crop for giving a better production. But once you are using a chemical manure, it is going to spoil only the field. So these are all the benefits of the biofertilizers over the chemical fertilizers. Once again, benefits of the biofertilizers continuing. Azolla, which is a biofertilizer, amends the soil with organic matter. Cyanobacteria in particular secrete growth promoting hormones like indole 3 acetic acid, indole butyric acid, naphthalene acetic acid as amino acids, proteins and vitamins to the soil, and so thereby it is able to improve the quality of the soil. The benefits of the biofertilizer is still it is continuing. Cyanobacteria grow well both in acidic as well as in the alkaline soils. Since cyanobacteria are potent naturalizers, they help in the naturalization of soil. The process of converting untenable fallow land to cultivable soil is termed as a soil reclamation. A very important concept, soil reclamation. While you are using a chemical manure, it loses the value and then it becomes useless. But once you are using a biofertilizer, the soil is reclaimed. It is brought to the live condition. So a soil for farming means it should be containing all the microorganisms. It should contain manure and it should be a potential soil. Then only it will be able to produce the crop which are growing over that. If it is a fallow land, If it is not containing any minerals, metals inside, it is not containing any microorganism, then it cannot support any crop growth over it. So the process of converting the untenable fallow land to cultivable soil is called soil reclamation, and that is done with the help of the biofertilizers. Blue-green algae play a vital role in this conversion. Then symbiotic nitrogen fixing rhizobium is a biofertilizer. It adds 50 to 150 kg of nitrogen to soil per hectare. Acetobacter and azospirillum secrete antibiotics which act as biopesticides. Heterotrophic mycorrhiza, which acts as a biofertilizer, increases the surface area of the roots of a host plants so that more absorption of nutrients by the roots is made possible. So by this way, all these microorganisms are able to improve the quality of their soil, thereby improving the quality of the crop also. So these are all some of the very interesting information that we have learned today about the food production. I have to sum up the whole thing. I am telling you that the food production is a very important, crucial problem today for our country, particularly for the country like India. And our scientists are constantly, regularly, they are trying all their level best to improve the quality of the crops, the production of the crop by different methods. And it is these different methods that we have seen: selection, hybridization, introduction, protoplasmic fusion, polyploidy, mutation breeding. By these different techniques, we are trying all our level best to improve our food production in our country. So it's a very interesting topic. Some of you will also be interested in that. After completing your course, you can go as a researcher, and as it is a very important topic concerned with our country, the food production, some of you will become interested and then do a further beautiful or research to improve the food value of our country. Thank you very much.